Think about this for a second. What powers a marathon runner, sprinting for the finish line? Or, hey, what about the very thought you're having right now? Turns out it's all powered by the exact same incredibly ancient chemical process. It's at work in pretty much every living thing on this planet. So today, we're going to break down glycolysis, the pathway that turns a simple sugar into the literal energy of life.
All right, let's jump right in. Here's our game plan. We're going to start with glucose, our main fuel. Then we'll walk step by step through the glycolysis pathway itself. After that, we'll see what happens to the stuff it makes, explore a really fascinating link to cancer, and finally, check out how other sugars get in on the action.
So let's kick things off with the star of the show, glucose. Now, we usually just think of it as a quick shot of energy, right? But inside our cells, its job is way more important than that. It's not just fuel. It's a fundamental building block for us.
I mean, glucose is unbelievably versatile. Its main gig, of course, is giving us that quick energy by getting turned into ATP. But it's also used for structure, kind of like the scaffolding that holds our cells together. If we've got extra, it gets packed away as glycogen in our muscles. Think of it like an energy savings account. And it's the starting material for other critical molecules, like the ribose that makes up the backbone of our DNA.
The name itself really tells the whole story. Glycolysis. It comes from the Greek words for sweet and splitting. And that is exactly what it is. It's a 10-step chemical assembly line that takes one glucose molecule, splits it right down the middle, and in doing so makes a small but crucial profit of 2 ATP molecules.
What's so cool about glycolysis is that it's the on-ramp for all energy production, whether you've got oxygen or not. If oxygen's around, it's just the first step in a super efficient process called aerobic respiration, which generates a ton of energy. But if there's no oxygen, it kicks off a totally different process called anaerobic fermentation. It's way faster, but a lot less efficient.
Okay, let's shrink down and zoom right into the cell to see this 10-step molecular dance up close. The whole thing is really elegantly split into 2 main phases. There's a phase where the cell has to spend a little energy, and then a phase where it gets a big reward.
You know the saying, you gotta spend money to make money? Well, that's exactly what the cell does here. In the first 5 steps, the preparatory phase, the cell invests 2 ATP molecules to kinda rough up the glucose and get it ready to be split. Then comes the payoff phase, the next 5 steps, where the cell cashes in big time, generating 4 ATPs for a nice little profit.
All right, so let's follow the money and walk through that initial investment. The whole goal of these first 5 reactions is to take one stable glucose molecule and turn it into 2 unstable, high-energy little pieces.
So the second glucose enters the cell, boom, the first investment is made. An enzyme called hexokinase grabs an ATP and slaps its phosphate group right onto the glucose. And this is a really clever move, because this new molecule, glucose 6-phosphate, is now negatively charged and can't fit back through the cell door. It's trapped inside. Next, the molecule just gets rearranged a little bit into its cousin, fructose 6-phosphate. Then comes the second and final investment. A super important enzyme called PFK1 uses another ATP to add a second phosphate group. Now we've got fructose 1,6-bisphosphate, which has a phosphate on both ends, making it really unstable and basically ready to explode. And here it is, the lysis in glycolysis. The unstable sugar is split right in half by an enzyme called aldolase. We get 2 different 3-carbon molecules. But here's the thing, only one of them, G3P, can move on to the payoff phase. So in step 5, the other molecule gets quickly converted into G3P. This is huge, because it means from this point on, every single reaction happens twice. The investment is done. The stage is set. Now for the really good part, the payoff phase. The cell is about to start making some serious energy from those 2 G3P molecules it just created. And the payoff starts right away. In step 6, each G3P molecule gets oxidized, and it hands off some high-energy electrons to a carrier molecule, NAD+, turning it into NADH. Think of NADH as a little rechargeable battery for the cell. Then, in step 7, an enzyme comes along and plucks a phosphate group off the molecule and sticks it onto an ADP, making our very first ATP profit. The next 2 steps are just some molecular shuffling. The molecule gets rearranged and has a water molecule removed, which creates an extremely high-energy compound called PEP. This is all to set up the grand finale in step 10. The enzyme pyruvate kinase swoops in, grabs that last phosphate from PEP, and hands it to another ADP. We get a second ATP molecule, and we're left with our final product, pyruvate. So let's do the final math. We spend 2 ATPs at the start, but because everything in the payoff phase happened twice, we actually made a total of 4 ATPs. So for every single molecule of glucose, the final scorecard is a net profit of 2 ATP, 2 of those energy-carrying NADH molecules, and 2 molecules of pyruvate.
But hold on, the story doesn't end with pyruvate. This molecule is now standing at a metabolic fork in the road, and where it goes next all depends on one single critical factor. Is there any oxygen around?
So let's look at the most common scenario for us, where there's plenty of oxygen to go around. This is the pathway that generates the vast majority of our cellular energy.
With oxygen present, pyruvate gets a ticket into the mitochondria. In there, it's converted into a molecule called acetyl-CoA, which then jumps into the Krebs cycle and the electron transport chain. This whole process squeezes every last drop of energy out of the molecule, generating around 30 times more ATP. It's a massive energy payout.
But what if oxygen is scarce? Think about a sprinter going all out in the last 100 meters, or a little yeast cell trapped in some bread dough. The cell has to switch to a different strategy to keep the energy flowing.
When there's no oxygen, pyruvate goes through fermentation. In our muscles, it gets turned into lactate. That's the stuff that makes you feel that burn during a hard workout. In yeast, it's turned into ethanol and CO2, the stuff that makes bread rise and beer bubbly. In both cases, the most important job is to recycle NADH back into NAD+, which lets glycolysis keep running and making at least a little bit of ATP.
Okay, now we're going to pivot to something really fascinating. We're going to see how this ancient, fundamental pathway has a surprising and very critical connection to cancer.
So back in the 1920s, a scientist named Otto Warburg noticed something really weird. He saw that cancer cells were just gobbling up glucose like crazy, but instead of using the super-efficient aerobic pathway, they were relying on glycolysis and fermentation even when there was plenty of oxygen around. It made no sense, and this paradox became known as the Warburg effect.
Let's just quickly remember how a normal cell works. If a healthy cell has oxygen, it will always choose the full aerobic pathway. It wants to get the most bang for its buck, the maximum ATP possible. It's all about efficiency. Cancer cells, though, they play a different game. They trade efficiency for speed. By putting glycolysis into overdrive, they not only get ATP fast, but they can pull molecules out of the pathway to use as building blocks for all the new lipids, proteins, and DNA they need to divide and grow like crazy.
Understanding this has opened up totally new ways to fight cancer. Some drugs are designed to directly block those key glycolysis enzymes. Others try to trap the lactic acid inside the cancer cell, which poisons it from the inside out. This effect is also exactly how PET scans work. They use a radioactive sugar that cancer cells love to eat but can't fully digest, so the tumors light up on the scan, showing doctors exactly where they are.
Okay, so we've been focused a lot on glucose, but obviously we eat other sugars too. Things like fructose and galactose are a big part of our diet, and our bodies have really clever ways of getting them onto that same glycolysis highway to be used for energy.
Let's take fructose, the sugar in fruit. It mostly gets processed in the liver. It gets a phosphate group added, then it's split into 2 pieces, and those pieces are quickly converted into the same intermediates we saw earlier, DHAP and G3P. It's like a different on-ramp that merges right onto the main energy freeway.
And then there's galactose, which comes from lactose, the sugar in milk. The enzyme lactase first splits the lactose into glucose and galactose. The galactose then goes through its own special multi-step process, but the end result is that it gets turned into glucose 6-phosphate, the exact same molecule from step 1 of glycolysis. So it jumps into the pathway right at the beginning.
Now, this is what's really interesting. It kind of reframes how we think about lactose intolerance. For most of the world's population, it's not really a disorder at all. It's actually the default setting. It's a normal evolutionary adaptation where our bodies just stop making as much lactase enzyme once we grow up.
Now, that is completely different from a much more serious, but thankfully rare, genetic condition called galactosemia. In this case, a person is missing a key enzyme for processing galactose. This means the sugar builds up to toxic levels in their blood. It's a really powerful reminder of just how critical every single one of these little molecular machines is.
So we've seen how just 10 steps can power a cell, fuel a disease, and even define parts of our diet. It is a process that is billions of years old, and it leaves you with a final thought to chew on. If this one single pathway is so central to life, disease, and evolution,
what other incredible secrets are just waiting to be discovered by studying the very core of our metabolism?